again, everyone. Today I'm going to show you the 3D model of my bacterial cell. This 3D model of mine was made by using wax paper and a little bit of clay, hence why the 3D model is just enough to fit in my palm. Unfortunately, I hold no footage of the process making of my 3D model. So feel free to just look and possibly cringe at the finished product. First of all, the definition of bacteria is that bacteria is a prokaryotic microorganism. It is a single-celled microscopic organisms that lack nuclei and other organized cell structures. Bacteria is the plural form of bacterium. While several bacterial species are pathogenic or capable of causing disease, most are non-infectious and many have critical roles in decay, fermentation, nutrient recycling, and nitrogen fixation. Now I will be discussing the parts of my 3D model. The red outer layer of my 3D model is the capsule. The capsule of most bacteria consists of a polysaccharide. The bacteria of a single species can be classified in different capsular serovars or serotypes based on the fine chemical structure of this polysaccharide. Its function is that it can attach itself to surfaces, it can protect against phagocytic engulfment, occasionally killing or digestion. It can also reserve of nutrients or protection against desiccation. The tail-like structure is called the flagella, singular for flagellum. The flagella is made up of a class of linear proteins called flagellants. The basal body transverses the cell wall and cytoplasmic membrane to anchor the flagellum and enables it to whirl about its axis like a propeller. Its function is that the flagella gives the bacteria the ability to move. The green area is the plasma membrane, also called the cell membrane. It is the membrane found in all cells that separates the interior of the cell from the outside environment. In bacterial and plant cells, a cell wall is attached to the plasma membrane on its outside surface. The plasma membrane consists of a lipid bilayer that is semi-permeable. The plasma membrane regulates the transport of materials entering and exiting the cell. These red dots you see on the 3D models are called the plasmid. A plasmid is a small circular piece of DNA that is different from the chromosomal DNA, which is found in all the genetic material found in an organism's chromosomes. It replicates independently of chromosomal DNA. Plasmids are mainly found in bacteria, but they can also be found in archaea and multicellular organisms. Plasmids have many different functions. They may contain genes that enhance the survival of an organism, either by killing other organisms or by defending the host cell by producing toxins. Some plasmids facilitate the process of replication in bacteria. Since plasmids are so small, they usually only contain a few genes with a specific function, as opposed to a large amount of non-coding DNA. Multiple plasmids can coexist in the same cell, each with different functions. This blue area of the 3D model is the cytoplasm. The cytoplasm, or protoplasm, of bacterial cells is where the functions for cell growth, metabolism, and replication are carried out. It is a gel-like matrix composed of water, enzymes, nutrients, wastes, and gases, and contains cell structures such as ribosomes, chromosomes and plasmids. The circles other than the red dots are the ribosomes. Ribosomes function as a workbench for protein synthesis, whereby they receive and translate genetic instructions for the formation of a specific protein. The yellow area of the 3D model is the cell wall. A cell wall gives the cell shape and surrounds the cytoplasmic membrane. It's an additional layer that typically provides some strength that the cell membrane lacks by having a semi-rigid structure. The hair-like structure that is attached to the capsule is the pili. The pili is an organelle of adhesion allowing bacteria to colonize environmental surfaces or cells and resist flushing. Lastly, this in the middle is called the nucleoid. The nucleoid is the space within a prokaryotic cell where the genetic information, called the genophore, is found. The nucleoid also doesn't take a uniform shape and has no specific size. That's all. Thank you all for watching.